So thank you for joining me today. My name is Tasmia. Hi, I am a third-year PhD student, as Juan just said. My dissertation project is a collaboration between the University of Calgary and the University of Alberta. So today I will be presenting some data from my PhD project related to the cognitive and neuroanatomical findings in children with ADHD. In terms of disclosure, I have no financial relationships to disclose. So here is just a brief outline of my presentation. I will be talking about giving you just an overview of ADHD, some of the behavioral and cognitive challenges that we see in the literature, as well as neuroanatomical changes observed. Next, I will be talking about the multimodal study we conducted at the Alberta Children's Hospital, briefly going over the methods, results, and discussions of our study. And finally, I'll end with the implications. So attention deficit hyperactivity disorder is among the most common neurodevelopmental disorder in childhood, with an estimated prevalence of 5%. Traditionally, symptoms of ADHD are characterized by developmentally inappropriate levels of inattention or hyperactivity and impulsivity. While it is common for young children to be active, energetic, and impulsive, children and adults with ADHD often have extreme difficulties in controlling their impulses and struggle to concentrate on monotonous tasks. These impulsive and inattentive behaviors affect other domains of their lives, such as their academic performances, their ability to maintain social relationships, and even motor impairments. A recent study in the United States found that parents with children with ADHD reported increased economic burdens, roughly spending 5 times the amount when they have a child with ADHD compared to a child without the diagnosis of an ADHD. So that's a big difference. In addition to their core symptoms, children with ADHD also face challenges with planning, staying organized, initiating tasks, shifting from one task to another, and monitoring their task performances. These challenges are categorized under the umbrella term of executive functions. While the literature indicates significant EF challenges in individuals with ADHD, there are inconsistencies, with some studies suggesting there is significant impairments, while others are showing more variable performances on these different neuropsychological-based executive function tasks. Additionally, behavioral challenges are observed in the classroom as reported by teachers. Increased aggression has been reported by parents. And then lastly, there is an increased rate of oppositional defiant disorder diagnosis in children with ADHD. Now, previously, ADHD was thought of as a behavior disorder. Then it became more of a cognitive-based disorder. Now we know there is a neurobiological link to ADHD. Now, for the current study, we were specifically interested to measure cortical thickness. Cortical thickness is a measure of the folding of the cerebral cortex. It could indicate maturation of the cortex, pruning, or even myelination. Research has suggested that cortical thickness can tell us whether where there is a trajectory of normal development, as well as if they see differences, it could indicate some sort of neuropsychiatric disorder or presence of some sort of neuropsychiatric disorder. In relation to ADHD studies, there has been research supporting delayed maturation of the cortex, specifically in the frontal areas of the brain, such as the superior frontal gyrus, as well as the anterior cingulate cortex. So for the purpose of my study, it was to better understand these executive function challenges reported by executive function challenges as we see in the neuropsychological-based performance task, as well as neuroanatomical differences in children with ADHD. Furthermore, we wanted to understand the correlations between the executive function performance task and neuroanatomical differences. So the study was part of a larger study, and today I'm only going to be talking about the clinical assessment and some of the MRI data. When the kids came to participate in the study, they actually completed 8 hours of work with me, but I'm only going to be talking about those 2 aspects. Moving along to my methods, we had 26 children with ADHD, and we had 24 healthy controls participate in our study. There were no biological, sex, or age differences between our participant sample groups. To be part of the study, our inclusionary criteria for the ADHD group was they had to have a confirmed diagnosis of ADHD from a healthcare professional. On the Conners parent rating scales, they needed to score greater than 65. Diagnosis was also confirmed through a semi-structured interview that I conducted with the child. And lastly, if they were currently taking stimulant medication, they had to be off for 48 hours. So we had a washout period. The children also completed IQ screener, where they needed to score a standard score greater than 80. And lastly, because our MRI was a core component of the study, they needed to be MRI eligible, so they couldn't have any metals in their body. In terms of the executive function task, we completed the digit span backwards from the WISC-5. They also completed the digit span, spatial span backwards from the WISC integrated. To measure response inhibition, we used the continuous performance task, 3rd edition, also the DCEP's color word interference task. And for response inhibition, I was specifically interested in that inhibition trial. Lastly, they completed the Wisconsin card sorting task to measure set shifting. Moving along to our demographic information. So as I mentioned, there was no age or gender differences, age or biological sex differences. There were also no significant difference in the measures of the 3 different measures we used for IQ. However, we did notice significant differences in the Conners inattention and hyperactivity subscale, which is what we expected. I also wanted to look at some additional demographic information. So our sample was not ethnically diverse. Specifically for ADHD sample, it was an 89% mostly white Caucasian. For their control group, we did a little bit better. However, I think it's important for us to acknowledge that we're not doing a good job representing and understanding some of the challenges for a more ethnically diverse population. We also looked at medication status. So about 84% of our ADHD sample were currently taking stimulant medication. No one in the healthy control group were on medication. We asked parents to indicate any comorbid diagnosis. So anxiety and learning disability were the 2 primary comorbid diagnoses we noticed. Lastly, we asked parents about their probable or confirmed diagnosis of ADHD in their immediate family. With immediate, we mean mom, dad, and the siblings. And what we found that about 69% of the immediate family had either confirmed or a probable diagnosis of ADHD in the ADHD group, while our control group only had about 26% of parents say yes to a family diagnosis of ADHD. So I think in the future, it'd be interesting to test this further. So moving along to my results. So for executive function performance, we expected to see differences across the board. However, the only difference we noticed was in the perseverative errors on the cognitive continuous performance test. All the other measures, they were pretty similar across the board. So for cortical thickness, we used the T1 weighted imaging, and I used FreeSurfer to do the cortical construction. And we also use quality control measures as indicated by the Enigma consortium to ensure that we were accurately measuring the thickness differences. Our hypothesis was to expect thinner cortical thickness in the superior frontal gyrus and the anterior cingulate cortex. So here's the superior frontal gyrus and the 2 region of the anterior cingulate cortex. So here are my cortical thickness results. So we only found significant differences in the right superior frontal gyrus. So there was thinner cortex for the ADHD group compared to our healthy controls. We did not find any significant difference in the anterior cingulate cortex. All right, so here is the medial and lateral view of the right superior frontal gyrus. We further looked at the relationship between cortical thickness and ADHD symptoms. So we found significant negative correlation between right superior frontal gyrus thickness and the ADHD subscales from Connor's parent rating scales. And our relationship suggested that 12.5% of the variance in inattention and 13.6% of the variance in the hyperactive impulsive scores were accounted by the right superior frontal cortical thickness. All right, so just my quick discussion regarding the findings. So we didn't see substantial neuropsychological based differences in our 2 participant subfield groups, except for perseverative errors. Now, perseverative errors could indicate challenges with inhibiting a previous response, or they were just impulsive and they weren't able to stop responding. It's also possible that we were just assessing a subgroup of children with ADHD who had minimal executive function challenges. It's not it's not unheard of that children with ADHD tend to perform better in a one-on-one setting, specifically in a clinical-based lab setting. We also had a small sample size because of the component of MRI being quite expensive. Lastly, our family, the families who participated in the study were generally on the high SES level. So that could also explain some of the differences. Now, these are just future research that I will be looking at, specifically the relationship with high SES. In terms of neuroanatomical differences, so we notice thinner cortex in the right superior frontal gyrus, as well as relationship with inattentive and hyperactive ratings on the Conners 3. Possible relationship could indicate that the right superior frontal gyrus could be a neuroanatomical target for future interventions. So moving forward, as I just wanted to indicate that the variable performance of executive function that we notice in our sample suggests that we need to understand how these tasks could be measured, or these performance could be measured in a more context-specific way. I know Maggie Toplak's lab is looking at an unstructured executive function measure, so it will be interesting to see what their results are showing. We were able to replicate previous findings for our neuroanatomical findings. And lastly, the relationship between executive function, parent ratings with cortical thickness would be something that we need to look at in the future. I want to quickly thank you, all my supervisors at the Alberta Children's Hospital and the University of Calgary, and my lab mates for their help to recruit all these participants for our study. Here are my references, and thank you very much.